Welcome to this session of Medicine in Briefs. In this session, we will discuss about CRISPR-Cas9 gene editing system. The history and application of the invention is fascinating. Let us dive in. In 1993, while the global scientific community was chasing medical gold, sequencing the human genome, a young PhD student named Francis Mojica was standing in the pink salt flats of Santa Pola, Spain, staring at single-celled microbes. He discovered a bizarre pattern in their DNA, short palindromic repeats separated by mysterious spacer sequences. For the next 10 years, Mojica studied these repeats with almost no funding. His peers called him crazy for wasting his career on what they assumed was evolutionary junk DNA. But Mojica was obsessed. Then came the sweltering summer of 2003. While analyzing those mystery spacers, he discovered they matched the genomes of bacteriophages, the viruses that infect bacteria. In a sudden flash of insight, Mojica realized this wasn't junk at all. The spacers were a genetic most wanted photo album, storing memories of past viral invaders to build adaptive immunity. But how did we get the name? By late 2001, different research groups were stumbling onto these identical repeats in different microbes. Because they weren't coordinating, the literature became a chaotic alphabet soup of names like SRSR, LERD, and SPIDER. To unify the science, Mojica wrote an email in late 2001 to Dutch researcher Ruud Janssen, proposing a single, elegant name: CRISPR, clustered regularly interspaced short palindromic repeats. The breakthrough began in 2011, when Charpentier identified tracker RNA in Streptococcus pyogenes, proving it was a critical molecular guide required by bacteria to cleave foreign DNA. She teamed up with Doudna, an expert in RNA structural biology, to recreate this system in vitro. Their landmark 2012 study demonstrated that the Cas9 endonuclease is directed to its target by a dual RNA duplex of CRAR and tracker. Crucially, they simplified this complex dual RNA structure into a single guide RNA, sgRNA. By doing so, they provided the global scientific community with an elegant, programmable genetic scissors capable of targeting and cutting virtually any sequence of interest. To understand how we deploy this tool, we must examine how bacteria use it to build adaptive immunity across three sequential phases. These are acquisition, expression and maturation, and interference. First, acquisition. When a virus infects a host bacterium, the host captures fragments of the invading viral DNA and integrates them as spacers in its own CRISPR locus, creating a permanent molecular memory of the pathogen. Second, expression and maturation. This locus is transcribed into pre-CRRNA. Tracer RNA then binds to the repeating segments, guiding the cell's endogenous machinery to process and mature these transcripts into functional Cas9 surveillance complexes. The final phase is interference. The matured Cas9 complex patrols the cell. Upon identifying foreign DNA, Cas9 scans for a short flanking sequence known as the PAM. Once verified, Cas9 unwinds the target double helix, base pairs with it, and introduces a precise double-stranded break to neutralize the threat. Today, we have bypassed the virus. By delivering this exact machinery ex vivo, we can target and disrupt erythroid enhancers like BCL11A to reactivate fetal hemoglobin, transforming fatal genetic disorders into manageable, curable conditions. Both Dr. Doudna and Charpentier were awarded Nobel Prize in 2020 for the discovery. Thank you for listening. See you in next session.